All right, biology students, we are going to get into protein synthesis and one of the central dogmas of biology. This was definitely a subject that when I learned more about it, my nerd senses were tingling because this was definitely something that I just thought was one of the most fascinating and just like coolest things, especially when dealing with any of the life sciences. So I'm going to continue to nerd out with protein synthesis. So, just a reminder that synthesis means to create or to make. So, we are going to be making proteins. So, remember that we have our DNA, right? That has to deal with our instructions. It's like a recipe book. And the recipe is going to make these proteins. So, RNA is going to help the DNA make these proteins. So, we kind of have this idea biologically, which means that there's this central dogma. So, we go from DNA to RNA to proteins, and that is called the central dogma. It explains how life kind of comes from DNA. So when you're dealing with DNA and it's just like an instruction manual isn't going to give you the product, right? You have to create or build. So if you ever go to IKEA and you get the instruction manual and you're like, great, thanks, but I can't build the whatever Swedish chair name that they give it without the proper materials. And that's where this comes in. So the central dogma is from DNA to RNA to protein. So how do we get from here to here? Well, we have to learn about our RNA. Okay. So a section of DNA that codes for proteins called the gene. So we learned about genes when we were talking about heredity and traits and that kind of thing. So the gene is read and the message is known or is used to make a protein. And these proteins then kind of determine the traits. And we're going to be doing an activity with this where you are going to go from DNA to RNA to protein to trait. So it's really cool. It's really cool how it works. And these proteins determine like your blood, like everything about you. So your eye color, your dimples, your hair color, your blood type, how tall you can be, how short you're going to be. Like it's so cool. All right. So protein synthesis is pretty complicated, but once again, this is just normal high school level biology, so we're not going to get too crazy into it, but definitely I'm just putting it out there. If you're going to take a science college class, this could be a cool one to do. So protein synthesis is the process of making proteins, right? So synthesis means to make, protein is what we're making. And there's two steps that happen, and these two steps are extremely important. So we have transcription, and this is when we get our mRNA copies from our DNA. And then the second step, we get translation. And this is where our ribosomes, remember those little organelles that look like Goombas from Mario or like weird hamburger buns? This is where they take that mRNA and that's where proteins are being made. So remember, ribosomes make proteins, and this is how they do it. All right. So transcription, this is the first step. So we go from and we're in the nucleus. So here we have DNA and mRNA, which remember is messenger RNA. So what happens? We have our DNA, it unwinds, right? Here we go. And the RNA polymerase, remember we had DNA polymerase when we were dealing with DNA replication. Now we're dealing with RNA because we need to make, now we're dealing with RNA polymerase because we need to make RNA. So we have this little RNA enzyme, little RNA polymerase, is going to come along here and use the DNA as a template to kind of make this mRNA, to transcribe it. Now that we have this copy of mRNA, it's going to get out of the nucleus. Okay. So a few things, and we'll do more activities like this with class. This is like bare bones explanation, and then together we'll go deeper into the subject. So we have DNA. We know the bases for DNA are A, T, C, and G, blah, 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 blah. Well, RNA has everything but no T's. So there's no T in RNA. So where you would normally put a T in DNA, we're going to put a U. And we're going to practice this a little bit because it seems to trip some kids up. But once you get the hang of it, it's really as easy as those base pair rules that we talked about in DNA. So same thing, right? We are making our mRNA, our messenger RNA, from our DNA. And the only big difference is instead of a T, we're going to put a U, which is uracil. Here she goes. So great, we got our DNA. Now we're at an mRNA. So now that mRNA leaves the nucleus, right, because we can't make our proteins inside the nucleus, we need to get to a ribosome. So we have our genetic code. We're trying to make a protein, right? So we have our mRNA. That message is in a language, and that language needs to be translated. So we transcribed it, right? We wrote down our instructions. Now we need to translate it. And to do that, we need to understand the code. And this is where we get to codons and codon charts. So this, if you're looking at this and being like, Phillips, what are you doing? Why do you hate us? I don't hate you. This is actually very simple to read. It just looks fancy. So we have our mRNA has codons. This is a sequence of 3 nucleotides that code for an amino acid. And remember, our amino acids are a building block for our proteins. So when we have our codons, we're going to break our mRNA into 3s. So if we have UAC, that's 3 bases, that's 1 codon. You can think of codons like words of the genetic code. So you're going to translate it a little bit at a time. So if I'm looking at my UAC and I got to use this wheel to figure out what amino acid it is. Oopsies. So we're going to go from start in the middle of the circle and work our way out. So they're saying it's triosin. I don't believe everything I read. I'm going to figure out myself. So we're going from the inside to the outside. So my first unit, my first letter, my first base is a U. So I'm only going to stay in this section. I don't have to worry about anything else. Then I'm going to move on to the second part of the wheel in the middle, and it's an A. Okay, so I go U, A. All right, and the last one is C. So I look at my last, stop disappearing. So I look at my last letter. Okay, there's C. So that tells me my protein. So we're working from the inside out. It's really not crazy. It's pretty straightforward. It just looks fancy. So then we have G, C, U. Okay, same thing. So G, C, and U. And notice that there are a bunch of bases or codons that code for the same protein. And that's really cool when we get into mutations. All right, kids, we're almost done, I promise. All right, so mRNA carries these messenger codons. rRNA makes up the ribosomes, which remember are these little organelles that make our protein. And then we have something called tRNA, and that matches the anticodon to the mRNA. So it brings the correct amino acid. They look like weird combs, and this is like molecularly what they look like. Very awkward. And we'll go over each one of those kind of like when we go through the process of the central dogma. So, second part, translation. It happens in your ribosomes. And what's involved is RNA, mRNA, ribosomal RNA, rRNA, and then transfer RNA, which is tRNA. So we are done with deoxyribonucleic acid. We're done with DNA when we get to translation. So there should be nothing to do with DNA anymore. So what happens? So our mRNA finds the ribosome where it needs to make these proteins. It binds and gets ready to get this tRNA going on. The tRNA brings the correct amino acid that matches with that codon, that 3-letter thingy. The amino acids link together to form this protein. Like this. All right, so we have our mRNA, we have our ribosome, we have our tRNA. So our tRNA is going to bring kind of the anticodon is what it's called. It's bringing the opposite of these 3 guys. So AUG codes for methylene 9, which is our start codon. And I'll talk to you guys about that later. All right. Codon, anticodon, and then it's going to go again. We're going to have this like weird pearl necklace thing going on of proteins. All right. So go ahead and practice. Because this is what, when you take your state test, this is really what they're probably going to be like swinging for. So we have DNA to mRNA to amino acid. So let's figure it out. So T is going to code for A. We would normally put T, but because we're dealing with RNA, we're going to put U, and then C and G. You go all the way through. And then we're going to use our codon chart. It comes in two flavors. It comes in circle and it comes in square. Whatever you feel more comfortable with is the one you're going to use. I teach both because I don't know what the state's going to give you. I learned on a square. I'm more comfortable with square, but there's a trend that the youths like the circle better, which is fine. Like I said, you read the thing, whatever shape it comes in. And here we have our amino acids, and we're going to do lots of practice with this. Whew, all right. Well, that was a lot of information, and we're going to take our time with this because it is a big thing of biology, and it's really going to help us get into our next unit, which is going to be evolution. So I will see you guys in class, and we will practice this more.